Good morning, everybody. We're still in the morning. It's lovely to see so many people here, and this is my first public event for Garvan. I came to work at Garvan only 3 years ago, and one of the things that I love about working here is that we work and breathe genomics and DNA, which is what makes us all the people that we are. And one of the things that's also lovely about working here is that everybody pulls in the same direction, and that's an important thing when you work in such a large organisation, that everyone who works here wants to make a difference in regards to suffering and the suffering that people with catastrophic diseases go through. So I will now put my timer on like Warren did, because Chris has put the fear of God into us about our timing. Anyway, so today I want to talk to you about our genome and how it has lifelong value. So Warren talked before about often when you have a genetic or a genomic test, that you can have tests at various times through your life. And what we're working on here is that we can actually take a person's genome and use Warren's fantastic data sciences platform and then dip back into it at various times in a person's life when they need it. And I'll take you through some of what we're doing in regards to that. But what I'd like to do is just to give you a very basic science lesson and just explain the difference between genomics and genetics, because people use those terms interchangeably, and look, they're just different, and it doesn't matter which one you use. But once upon a time, we just used to be able to study 1 or 2 genes at a time. Whereas now we can actually look at a person's whole genome, which is their whole genetic makeup. And our genome has variation in it. Otherwise, if it didn't have variation, we'd all look the same. So the variation gives me curly hair and freckles and makes me 5'7", and the genome in yours makes you taller than me. You've got different skin and whatever. So we have normal variation, but it's the variation that causes disease that we look at here at Garvan. So some of our variation in our DNA can cause us to develop diseases. We have this technology that we call next-generation sequencing, and that's been a huge breakthrough in studying our whole genome rather than just genes at a time. And here at the Kinghorn Centre for Clinical Genomics at the Garvan, we have the world's most advanced high-throughput sequences, which can sequence thousands of genomes at the one time. And we feel very proud of that because it was the first accredited laboratory. So it means that we can offer genomic testing to people in a safe and ethical way that's in a regulated environment. So if we think about our current understanding of the genome, as Warren said before, the cost of genome sequencing has actually decreased enormously. And we now have thousands of genomes here at Garvan that we store, and we store them for a lifetime. But what we know about the genome is still relatively small, and that's the challenge that we face every day. So if we think about this is what we currently know about the genome, there's so much more that we need to understand in regards to our genome and the diseases that it causes and the way in which those diseases occur. And that's currently what we're actually working on, is what we call bridging the gap. So bridging the gap about what we don't know and bringing more understanding so that that can help people in regards to either knowing that they've got an increased risk of developing a disease, so we call that disease predisposition, or actually treating a disease. So one of the areas that I'm particularly interested in working in is identifying people who have a higher risk of certain diseases and then doing things to actually prevent that from occurring. So what can you do with a sequenced genome? Warren talked before about how he's got this platform that holds all these genomes. There's many opportunities during our lifetime to actually go through and use our genome. So the good things are that you only have to be tested once, and we can sequence a genome and then we put it away in a very secure place, and if you need it again, we take it out, we reanalyse it, and we give more results. So we can reuse the data over and over again, and we can reuse it for an individual's health, or we can actually use it as aggregated data to do research on to find new cures for diseases. But we have to store it securely, and we have to store it privately to make sure that it's used in the best way. So privacy and security are actually paramount to us, and particularly the issue of trust. So I just want to take you through, I'll just use my big girl voice, the lifetime of the genome, which actually correlates really when you think of what we call the life cycle. So I'm also a family therapist, and we talk about this life cycle of people's lives. So you meet someone, you date, and then you decide that you're going to get together with them and have a family. And so then we think about this reproductive carrier screening. So that's when you test two people to see if they carry genes that might cause a problem in their children. And then you go through life and a pregnancy is achieved, and you can have testing during pregnancy to see whether that baby may have any problems. That baby is then born, and you can test the baby, its genome, to see if it might have any diseases. Then we move on, and the person gets older, and they may have developed conditions themselves, and so we can use their genome again to see if we can diagnose that condition, and not only diagnose it, but use it, find a personal treatment. And we also can use a person's genome to see if they might carry genes that cause a predisposition to disease. And we can use a person's genome to see how they respond to medications. So your genome can actually influence how you respond to drugs. Mandy's nicely talked about how you can use a person's genome in regards to either helping to diagnose cancer or plan a person's treatment. We can use a person's genome in regards to helping fight infectious disease. And we can also use a person's genome in the post-mortem phase to actually find out what was the disease that they died from, and is that something that their family may be at risk of. So this is what we're working on now, is actually thinking about the genome in quite a different way. How can we use the value that's in our genome to actually inform not our own health, but future health? So I'm going to go faster because I know I'm going to run out of time. So we have this thing a program in KCCG, which is called the One program. And basically what it is that we want to use people's genome for a lifetime. So we're actually we have our first group of secondary school students who have given us their samples, and we are looking to see if they might carry genes that when they plan children, that those children might have an increased risk. And then we are going to lock their genomes away in what we call the gene trustee, which is a bit like a bank vault. And 3 people will have access to that genome, of which the person whose genome it is will have a key, and 2 other people will have a key. So it's very trusted, it's very secure, and they can have life access to their genome along that path. And so we're building the framework to enable this to happen. And there's one gentleman who I work with, Professor Leslie Burnett, who is the brains trust behind all of this. And he's a real leader in this world in enabling us to build this framework. So it means that we'll put people's genomes away, and we'll take them out of the vault, and we'll reuse them for the purposes of disease throughout their life. So Lifeaxis, what are the things that some people want to know as they get older? So we've got these secondary school students' genomes at the moment. They might come back to us when they're 40 and say, I want to know if I've got that gene that Angelina Jolie's got. Do I have BRCA1 or 2? What's my risk of getting breast cancer? And so we can tell them that. Or they might say, I'm about to start a new drug. Can you tell me if my genome will affect the way I process that drug? So these are the types of things that we're starting to work on now in regards to actually preventing disease. And so one of the main benefits in thinking about this is how can we benefit patients? So we've heard all about the science, and Mandy talked about the patients that they've been helping. So the group I actually lead is called a clinical translation group. How can we take that not only to the bedside, but how can we take that to communities of people so that they can benefit from genomic information? So just to go back and think about how things are inherited, we know that someone like Angelina Jolie has a condition that's called a monogenic condition. One gene, very big effect. So she has this gene variation in her BRCA1 gene, gives her a very high risk of developing breast and ovarian cancer in her life. She's taken measures to reduce that risk. So it's one gene, and you can see that age is not such a big factor, environment and lifestyle are not such a big factor. It's the gene that's the biggest risk for someone like her. But for most of us, we don't inherit one gene. We inherit many genes, and each can have a small contribution to risk. And that's how the work that we're doing at the moment is focusing on. It's called polygenic risk, many genes that can cause an increased risk. So you can see in the diagram here, the little green ones are our genetic risk, what we're born with. Age is in the blue, and environment and lifestyle is in the mustard colour. So many diseases are caused by this type of inheritance, and particularly things like heart disease. It's a very common disease in our community. And also most forms of breast cancer are caused by this type of inheritance. So what it is that you're actually born, your parents pass on the DNA, they pass on a few small genetic risk factors which slightly increase your risk. You go through life, you get a bit older, and as Warren said, you might eat too many McDonald's hamburgers and have too many fries and Coke. You age a bit more, you eat a few more McDonald's fries and Cokes, and then you get a bit older and you've really eaten too many McDonald's fries and Cokes and you actually develop the disease. Diabetes is a classic example of the way this works. So if you think about diabetes and what you know about diabetes, you can be born with a slightly increased genetic risk to develop diabetes, and then as you get older and you eat lots of McDonald's, your risk increases. You can actually change your risk of diabetes. You can't change your genetic risk, but you can do things to alter your risk by losing weight, not drinking so much alcohol, not eating all those McDonald's and fries. So they're the things that you can do. You can't change that baseline genetic risk that you were born with. You can't change your age, but you can change your diet and lifestyle and environment. So I talked briefly about pharmacogenomics. So pharmacogenomics is where we are doing DNA tests that can inform what drugs people are prescribed and also how much they are given. So if you think about a common drug like codeine, if you give some people codeine, it has no pain relief effect. You give it to other people and they're almost unconscious. So why does that happen? So part of that has to do with the DNA variation that we have. We all process drugs differently. And so we're now working in this area of doing what we call pharmacogenomics to help when prescriptions are written so that they're also personalised to that person's genomic variation. So here we are back in what I call the cycle of life in the genome. And you can see again, these are all the programs that we're actually working on here at Garvan in regards to using a person's genome for their lifetime. So one of the other things that we're working on is that we can do research in looking at a person's genome for their lifetime, and we find a lot of information from that genome that is actually beneficial to the person themselves. And we call those clinically actionable results. So these young secondary school students will look at their genome and we might find over time that they have a gene that might cause a predisposition to prostate cancer if they're a male. And so one of the things that's important to do is to return that research finding to that person so that they can actually use that information. Lots of people participate in research. There's lots of information that can go back to clinically benefit people. And so we're actually setting up a genomic resource centre. I'll go forward, which will be staffed by genetic counsellors to actually help research participants in determining is that information beneficial and would they like to know it. So that's really taking the research information to the benefit of the research participant. I just want to go back and briefly touch on what we know about the psychosocial implications of genetic information. So for a long time, people have thought that genetic information could be harmful. Having worked in genomics for over now 30 years, I might say, we actually don't know that there's any harms with genomic information. People get distressed, but it's not harmful in the long term. In fact, people talk about the value of getting genomic information, and its value includes that it provides an explanation as to why something has happened. It actually helps that person make meaning of where their situation is. They can plan for the future, they know what the cause of the condition is and they can plan for the future. And also, if it is an inherited condition, they can actually prevent future disease in their family. So rather than thinking about harms, we now think about value of genomic information. And rather than thinking about concerns about privacy, insurance, all those sorts of things, we are starting to really realise the great value that genomic information can have. I think I'm just about out of time. So thank you all very much for coming, and I've got 15 seconds left, and there we go. Thank you.